will present the introduction to polysomnography, a little bit about sleep physiology, and the basics of a polysomnogram setup. The objectives of this presentation include the discussion of the importance of sleep and sleep hygiene for overall metabolic and physiological health, an outline of the basics of sleep physiology, and we will describe the basic components and setup of a polysomnogram. All right, let's start by talking about the purpose of sleep. Now, obviously, sleep serves many purposes, but we'll just talk about a few of them. Energy conservation is on the top of the list. So during sleep, metabolism is decreased for the purpose of allocating limited energy resources to other things that need to happen during the sleep cycle. The molecules that you see up there, the one on the left represents ATP, which you probably know is the main molecule used for energy in the body. As those phosphates are cleaved from that ATP molecule, eventually it leads to just adenosine, which is the molecule on the right. The higher the adenosine molecule concentration is in your body, the more likely you're going to want to go to sleep. So high adenosine concentration is very much associated with sleepiness. It's very interesting. Now, the energy that we do have in there needs to be used for cell repair. And in order to repair cells or anything that has gone wrong in the body throughout the day, there must be protein synthesis. And along with that protein synthesis will be growth hormone function. So growth hormones play a huge role in repair in the body. And you've learned before about the central dogma, where we have the DNA, which is basically our script for everything that happens in our body, and how that DNA is transcribed to mRNA, messenger RNA, and eventually that messenger RNA will be translated to a protein. And protein is what really serves the function of everything in our body: enzymes, channels, signaling molecules, etc. Also during sleep, there's a huge promotion of immune function. So that is a time when the body is recognizing what is good and what is bad, what is self and what is not, and how to deal with that in a healthy way. People who get adequate sleep tend to be more immune to diseases and get sick less because they have optimal immune function. And then also during sleep is when we experience the most important function in regards to brain activity and neuronal activity. And this activity is really about maintaining homeostasis and developing what we call brain plasticity, which allows a person to be able to adapt to changes in their environment, changes in their situations. And this brain plasticity also plays a role in memory consolidation. So throughout the day, you're exposed to a lot of stimuli, which produces a memory, whether you're aware of it or not. And at nighttime, when you're sleeping, well, assuming your sleeping is at nighttime, while you're sleeping, your neurons will undergo changes, kind of cleaving of the information that doesn't need to stay there, but consolidating the memories that do need to stay there. So for example, when you study all night long before an exam, you're not likely to do as well because you actually didn't take the opportunity for that memory consolidation to occur. And sleep is also important for neuronal maturation. So many of you probably noticed that infants will sleep a lot during the day, and that's because sleep is required for neuronal brain maturation. When we look at this picture below, on the left side you see 4 neurons, A, B, C, and D. And this represents a healthy neuronal signaling pathway. So you can see A is communicating to C, and the communication is occurring through those signaling molecules. You can see them as circles between the neurons. And B is healthily communicating to D. But if during the day something happens to B, causing it to be sick or not functioning right, you'll notice that the signaling between B and D has ceased. Well, during sleep, the neuronal plasticity and the repair processes allow the conduction to actually resume, but just through different pathways. So you can see the bad neuron has now been eliminated, and A can now effectively communicate to C and D. So this is just a very simple example of what your brain is doing while you're sleeping. Now, sleep is regulated by the circadian rhythm, which is a daily cycle of biological activity that is very much influenced by variations in the environment. This particular rhythm oscillates every 90 minutes while awake. Factors that affect the circadian rhythm are light, temperature, food or biochemicals, drugs, and activity. Light is very important, and the sleep-wake cycle is very, very sensitive to light. When the retina recognizes light, a neuropeptide called hypocretin, which is also called orexin, is secreted, and this stimulates the wake cycle. Temperature and thermoregulation. Higher temperatures tend to promote sleepiness, but lower temperatures might maintain sleep. So some of you might notice that when it's really hot outside, it's harder to sleep throughout the night, but you do get sleepier earlier. Certain foods will contain biochemicals that promote sleepiness and vice versa. Drugs the same way. There are drugs that someone could be taking that promote sleepiness or promote awakeness. And activity. Exercise tends to create wakefulness, but exercise during the day tends to allow for healthier sleep throughout the night. So this figure represents the circadian rhythm, and if we start at 12 a.m. on the top, assuming that somebody is asleep at 12 a.m., when they get to about 2 a.m., this is when they're experiencing the deepest sleep. And when we talk about deep sleep later, we're usually talking about sleep stage 3. As time goes on, about 4 a.m., that's when the person is experiencing the lowest body temperature. And if you ever dream or you remember your dreams, you might notice that you will recognize those dreams in the early mornings around 4 or 5 a.m. It's a very interesting phenomenon. About 6:15, this is when people will experience the sharpest blood pressure rise. At about 7 a.m., melatonin secretion begins to stop. Now, melatonin is very important for initiating sleep or that feeling of sleepiness. So this is when most people begin to wake up naturally. At 10 a.m. is when people will experience their highest alertness. You guys have probably noticed that you're probably most productive in regards to work around this period of time. Around 2:30 p.m. is when people might experience the best coordination of their muscles. Around 3:30 p.m. is when people might experience the fastest reaction time. And then 5 p.m. is when you want to get your exercise because you usually will have the greatest cardio and muscular efficiency. But at 6:30 p.m. you'll start to develop a little higher blood pressure and your temperature starts to go up about 7. And at that period of time, you're starting to undergo the changes that are associated with the light to dark changes in the environment. And around 9, most of the time at 9, it is dark outside, and this is when melatonin will begin to kick in again. Now, sleep disturbance is actually not uncommon, and there are a lot of different types of sleep disturbances. In the figure to the upper left-hand corner, these are disturbances specifically related to sleep. So some examples that are up here are insomnia, apnea, narcolepsy, which we'll talk about, restless leg syndrome, disturbed sleep from other reasons, children, and some issues that are not necessarily reported. Now, along with sleep disturbances or sleep problems comes comorbidity issues, and these are the statistics that are associated with comorbidity with sleep apneas or sleep disturbances. And most people that do have sleep disturbances, specifically sleep apnea, they also have heart failure or chronic lung disease, hypertension, coronary artery disease like atherosclerosis, diabetes, renal disease, and erectile dysfunction. All of these are related to inflammation, oxidative stress that alter metabolic function. Most adults require about 8 to 8.5 hours of sleep a night. that's a requirement. Doesn't mean that's what we're getting. And unfortunately, because a lot of us don't experience the number of hours we need and the quality of sleep that we need, sleep problems have actually added up to about 15.9 billion cost to the national healthcare bill. That's a pretty big number. And overall, there's about 84 classifications of different sleep disorders. So what is sleep apnea? Well, apnea means to not breathe, to stop breathing, absence of breath. And there are three main types of sleep apneas. One, obstructive sleep apnea, or what we call OSA, means that there's an effort to breathe, but there's no flow. And this is usually due to an occlusion of the upper airway. Now, when we're looking at a polysomnogram, this is a very simplistic version, obviously. Three important parameters we will look at will be nasal flow or nasal pressure, and this is generated by a nasal cannula in the nares. Chest belt effort, and this is generated by a piezoelectric belt that is wrapped around a person's chest. And abdominal belt effort, and this is also a piezoelectric belt that's wrapped around the abdominal area of a person. So if we look at this OSA figure, the nasal flow pressure, you can see there's a blip that goes up and then it's a flat line. That means there's no airflow. But if you look at the chest and abdominal belt, you can see squiggly lines, and each one of those squiggly lines going up represents the person trying to initiate a breath. So their chest and abdomen are moving, but there's no flow. This indicates an obstruction to flow, an occlusion in the upper airway. And then we have central sleep apnea, or CSA. In this situation, there is no effort, and because there's no effort, there's no flow. So if we look at the nasal flow or pressure waveform, you can see there was a breath, but then it flattened out. That means there was no flow, and that no flow was accommodated by no chest or abdominal belt effort. So the person wasn't trying to breathe. This means that their brain did not try to initiate a breath, and this is what we call central sleep apnea, central coming from the brain. And then there's a combined or mixed sleep apnea, and it almost follows this particular path or order. We will see central sleep apnea followed by obstructive sleep apnea. So if we look at the nasal flow / pressure waveform, you could see that there's no flow. The flat line is there. In the chest, the first half of that apneic period, there's no chest or abdominal belt effort. But as we get closer to the end of that apneic period, the latter half of it, you can see there's movement in the chest and abdominal belt. So this represents central sleep apnea followed by obstructive sleep apnea in the same apneic period, and this is what we call combined or mixed sleep apnea. So how do we know somebody has sleep apnea without testing them? They tend to have similar signs and symptoms. So first of all, they will often report that they are not sleeping well, that they have insomnia, but they may not be able to define why that is or describe why that is. They almost always will experience snoring, and because of that and the gasping that happens during the night, they almost always will experience a dry mouth or throat in the morning. They also tend to have headaches in the morning. They're more likely to experience memory loss. They're more likely to have attention deficit issues. They're more likely to experience depression or be moody, fatigue, having to urinate a lot at night, and if it's a male situation, they might also experience impotence. Okay, so moving on, just adding a little bit of the basics of physiology. An important part of the sleep-wake cycle is the RAS, the reticular activating system. This system is what's responsible for arousal from sleep, wakefulness, and attention. It is a component of the reticular formation in the brainstem. It contains neurons that produce a network that project to the anteriorly to the hypothalamus, posteriorly to the thalamus, and directly to the cortex. There are four main components of the RAS that are discussed in more detail in the Physiology of Sleep lecture that I will add for you guys. There's a lot of detail that goes into it, so I'm not adding that here. But listed right below it are the main components of the RAS. The RAS is activated by the lateral hypothalamus, which releases a neuropeptide, orexin, which we talked about earlier, in response to light hitting the eyes. When this happens, this is when arousal is stimulated and the transition from sleep to wake occurs. Now, the RAS is very interesting because the RAS basically takes the information from the systemic system, the environment, transmits it through the thalamus to the rest of the brain so that your brain can interpret it and come up with a plan of action that it sends back down to the body. Now, along with the RAS is what we call the suprachiasmatic nucleus, or the SCN. This is located in the hypothalamus, and this is responsible for regulating the circadian rhythm, specifically the circadian rhythm as it relates to sleep-wake cycles. So it's often referred to as the circadian pacemaker, and it's very, very responsive to light. So if you look at this figure over to the right, you can see an eye, and you can see how that eye is communicating signals to the hypothalamus. And you can see the hypothalamus in the area that the red arrow would be pointing. And I've put a picture below it kind of representing the structures that would be within that hypothalamic area. And you can see where that suprachiasmatic nucleus would be located there. It's circled in red, the circadian pacemaker. And then you can also see the reticular formation there in the brainstem. That is representing our RAS. So the RAS is taking these signals, communicating them to the brain, but the RAS and the SCN will work together to regulate the sleep-wake cycle and how responsive a person will be to that sleep-wake cycle. Now, there's some chemicals that are very much involved in circadian rhythm or sleep-wake processes that activate the cortex to give us our wakefulness. And those are dopamine, acetylcholine, noradrenaline, histamine, and glutamate. And again, I talked more about those in the Physiology of Sleep PowerPoint that I will also be adding. So during sleep, the brain manages to cut off sensory input. And that means the RAS is not doing its thing. Yet it still can let in really important stimuli, so it's not completely paralyzed, if you will. No sensory information is going to get up to the cerebral cortex without passing through the thalamus. So the thalamus is very, very important here. If the gate of the thalamus is closed, gate is basically what we'll call a neuronal pathway, then the cortex can shut out the world and go into sleep mode. And this is the same thing that happens when somebody is in coma. Now, the ascending pathways here originate from a group of neurons around the fourth ventricle and the rostral pons near the midbrain. And this is what we talked about in the previous slide. Most of these neurons are acetylcholinergic, this is very important, and project to the thalamus, controlling whether or not that gate is open or closed. So it's the acetylcholinergic neurons that really play a huge role in determining awake or sleep state. So what does acetylcholine do? Acetylcholine sensitizes the neurons of the thalamus. So what this means is it lightly depolarizes the thalamic neurons. It does this by closing a hyperpolarizing potassium channel. Then the assisting system can make the thalamus more sensitive to sensory input. This situation should correspond to awakeness, alertness, being aware of what's going on. Now, as mentioned in the previous slide, the acetylcholinergic neurons project to the sensory areas of the thalamus. and into what we also call the reticular nucleus, which is a layer of cells that wrap around the thalamus like a rind of an orange. So this here, the reticular nucleus here, is different than our reticular activating system, even though it has a similar name. In this situation, the reticular nucleus of the thalamus has a general inhibitory effect on the sensory thalamus. So when it's active, it's inhibiting the thalamus. So when it's active, it's not allowing signals to go through the thalamus, which means we're asleep. The sensory thalamus is sensitized by acetylcholine, or facilitated, if you will, as described previously. The reticular nucleus is inhibited by acetylcholine. So you can see how acetylcholine plays a role in inhibiting or activating either the reticular thalamic nucleus or the thalamus in order to allow sleep or wake to occur. So what's happening in the thalamus and the RAS during awake? Well, the cholinergic cells are active. These are the acetylcholinergic cells. They're going to facilitate sensory thalamus activity, but they're going to inhibit the reticular nucleus activity. The inhibition of the reticular nucleus actually excites the sensory thalamus as well, so it's got a double whammy there. And the excited thalamus lets all sensory information through. This produces strong desynchronized activity in the cortex due to all the input. And this is wake. This is what we call wake. And what about sleep? Well, the RAS, the cholinergic pathway here, is underactive, and therefore the sensory thalamus is not very sensitive. The reticular nucleus is freed from inhibition, so it now can inhibit the sensory thalamus. So the net effect is that the thalamus is very insensitive to sensory stimuli because the gate is closed and the cortex can rest. When the thalamic neurons are hyperpolarized, they have slow intrinsic waves of activity, which may be the source of the slow synchronous patterns that we see on the sleeping EEG, and we'll talk about that another time. It also may function to keep the cortex in shape, ensuring that even when you are asleep, the cortical neurons are still active. Okay, so sleep stages. I'm only going to talk about 4 here. And we'll get more into detail about different sleep staging later. But for now, let's talk about stage 1. This is also referred to as N1 or NREM1, non-REM 1. So this is stage 1 sleep. It takes up about 5% total sleep time. It lasts about 1 to 7 minutes. It's a very light transitional state of sleep. In stage 1, a person is very easily aroused. They might have myoclonic jerks. You guys know what that is, where you start to fall asleep and then you kind of jerk and wake yourself up. There can also be hypnagogic imagery or daydreaming. Sometimes if somebody is awakened during stage 1, they will experience retrograde amnesia. And the quantity of stage 1 sleep directly correlates with daytime sleepiness and subjective refreshing sleep. So somebody who's in stage 1 sleep more than they should be is not going to get that restorative sleep, and they will recognize that as feeling worse during the day. Now the brain waves that are present during stage 1 sleep are what we call theta waves, and we'll talk more about that another time, but that gives you an idea of what they look like there. When we look at the eyes, we've got an EMG on the eyes, EOG actually on the eyes, where we're looking at the electrical activity of the eyes. You can see that there's a little bit of rolling that happens, but not a lot of movement. And then when we look at the chin is very, very tight. That means that they're not in a deep sleep. They're definitely not in a REM sleep, but you could see a lot of movement there. It's not an up and down movement per se, but it's just it shows the the contraction of that muscle, the lack of relaxation. And then we have stage 2 sleep, which takes up about 50% of total sleep time and lasts about 10 to 25 minutes each time, and it is also considered non-restorative sleep. Now, the brain waves that you'll see in stage 2 are also theta waves, but also we will see sleep spindles, which will be really, really fast waveforms, or K complexes, which are going to be wider. And again, we'll talk about this more a little bit later. This is just a brief introduction. And when we look at the eyes, we don't see a lot of movement there, and the chin still has that tautness. It's not completely relaxed. Moving on to stage 3, or non-REM 3. This is what we call our deep sleep. There is a high arousal threshold, so it's harder to wake people up. This is where the body will experience most of the musculoskeletal recovery. It occurs most frequently during the first to third of the night, and it takes up about 20% of total sleep time. This is where dreams and night terrors will occur, but you won't remember them. The brain waves that we see here will be slower, and we call those delta waves. The eyes might have a little bit of movement, but not significant. But the chin is still taut. We still have a lot of tenseness in the chin. And then when we get to REM, REM is rapid eye movement sleep. When you get to see this rapid eye movement on an EOG, on a polysomnogram, it's pretty interesting. It doesn't happen all the time, but when you get to see it, it's pretty cool. So the eyes will move back and forth in a coordinated way. You see the chin at the bottom is very flat, so the person has experienced a state of atonia. That means their muscles are basically paralyzed, so they can't move or act out any dreams they may have. REM takes up about 20% of total sleep time, and it lasts about 1 to 5 minutes. And it occurs more frequently the latter third of the night, usually. This is where psychological restoration occurs mostly, and this is also when apnea will occur mostly because of that state of atonia. People will experience dreams and nightmares here, but they will almost always remember them. Now, the brain waves that we see are still called theta waves because the theta is a particular frequency, but sometimes they will have a more sawtooth configuration. And below that, you can see that coordinated eye movement that happens. So this is just a very basic introduction to sleep stages. We will get into this in a lot more detail later. So you probably noticed that I mentioned stage 1 takes up a certain period of time, stage 2 takes up a certain period of time, etc. Well, those periods of time all are supposed to occur within a 90-minute period of time, a 90-minute cycle. So during wake time, we've got 90-minute circadian cycles. During sleep time, we do too. And it's good for that transition to occur without disturbances. But when there are disturbances throughout the night, we call that sleep fragmentation. And people that experience more sleep fragmentation are people that will experience more daytime sleepiness. Sleep fragmentation is characterized by repetitive short interruptions of sleep that a person may or may not recognize as happening. The number of arousals or fragmentations are related to the total sleep time, and this is useful to quantify for sleep fragmentation. And we'll talk about that a little bit more in the next slide. So this is a classic epic that we would see on the screen when we're looking at a polysomnogram while a person is sleeping. So the first 4 rows, C3A2, O2A1, ROCA1, LOCA2, these are EEGs. So we're looking at the brain waves, the electrical activity of the brain through different leads. And we'll get into this in more detail another time also. And if you notice in the very top one, the C3A2 lean, you can see a spike that is circled in red. That indicates an arousal, and you can see two more at the end. That means that something stimulated the brain to say, hey, something's not right, maybe you need to wake up, maybe. And if we go down to the fifth line, where it says Chen EMG, you can see that the person is having changes in their chin tightness, if you will. There's more muscle activity. And it seems to be occurring probably with a snore. But you can see that those chin EMG changes also occur around the same time an arousal in the EEG is occurring. Now the sixth line is the ECG, and we won't talk about that right now. We'll talk about that later. And then below that we have a right leg EMG and a left leg EMG. So we're looking at the muscle activity in the right and the left leg. And you can see that the right and left leg both move when that arousal and that snore is happening. So this is an example of fragmentation. Well, where did that come from? Well, if we look down, you can see where it says nasal or oral airflow, chest movement, abdominal movement, and circled in red, that big circle, you can see that the nasal airflow decreased. And it looks like the chest and abdominal movement changed also. So when you see a decrease in airflow like this, it's not absent, so it's not an apnea, but it is lower than it should be. They call it hypopnea. So in this situation, there was an obstructive hypopnea that the brain recognized as a problem, and that caused that arousal in the brain. So this is just a basic example of what we would see on an epic if we got to see an arousal caused by a respiratory related problem. In this situation, the respiratory related problem is an obstructive hypopnea. So why do we care about this? Well, snoring, first of all, is not benign. So we do want to do sleep assessments on people who snore. Snoring means that there's poor airflow. Apnea obviously means that there's no airflow. Now, when there's low or absent airflow, this means that for a brief period of time, there's also going to be lower oxygen and higher carbon dioxide because the ventilation going through the lungs is not normal, it's decreased. When the oxygen goes down and the carbon dioxide goes up, this is going to cause a nervous system arousal. And this also causes fragmented sleep. This arousal is going to stimulate a sympathetic response, a stress response. And this is going to cause an increase in adrenaline, or what you guys know as epinephrine, and cortisol, among others. So if this is happening throughout the night, you can imagine somebody's trying to rest, they're experiencing a stress response, and they're having the sympathetic response increase these neuronal hormones that actually can cause changes in their metabolic health. So for example, people that have a lot of fragmented sleep apneas, etc., tend to have an increased blood glucose level. Well, when cortisol is released, it also increases blood glucose because you need glucose for fight or flight. That's the sympathetic response. Well, over time, this can result in diabetes mellitus type 2 and vascular disease like atherosclerosis. These people will also experience higher blood pressure because of the sympathetic response. This can lead to hypertension, which can lead to headaches, which over a long period of time can lead to stroke and vascular disease. Now, this change in epinephrine and cortisol levels throughout the night also alters fat metabolism, ultimately resulting in increased visceral, or what we call organ, or what we call central fat. So visceral adiposity is the bad fat. There are people who can be fatter than what we think they should be, but they don't have a lot of visceral adiposity, so the fat that they have is not unhealthy. This is the unhealthy type of fat, because the fat cells become hypertrophied, they become bigger, and as they become bigger, they become basically little inflammatory organs releasing inflammatory cytokines, etc. This predisposes people to diabetes and vascular disease, among other things. And then, of course, there's going to be altered central nervous system signaling. This, over time, can result in depression, irritability, attention deficits, poor cognition, memory, and learning. So what do we do about this? Well, sleep hygiene is very, very important. And there's a lot of people nowadays who have a light source where they're sleeping, their cell phone, a TV, maybe even an alarm clock. It's important to realize that light disrupts sleep by increasing that orexin, or the hypocretin that we talked about, that promotes wakefulness. So you want to make sure that the light sources are not in your view when you're trying to go to sleep. Eating lighter in the evening is better. The heaviest meal should be lunch. It's not uncommon for people who eat heavy at nighttime to not be able to sleep as well and develop GERD over time. And of course, chemicals like caffeine, alcohol, nicotine, etc. can inhibit good sleep. Now, a lot of people believe alcohol helps them sleep, but actually alcohol decreases your ability to get into REM sleep, which is that sleep needed for psychological healing. Heavy exercise, you shouldn't be doing that before 2 hours that you want to go to bed. Instead, you would want to do yoga or some kind of meditative exercise that kind of relaxes you. Work planning should not be done in bed. Your bedroom should be different from the rest of your work activity. It should be a time or a place where you can actually relax. And make sure your sleep area is conducive to sleep. So make sure you don't have too much noise, too much light, make sure it's comfortable, etc. And go to bed when you're sleepy. You know, don't try to stay up too late because you have to, and don't try to go to bed when you're not sleepy because then you'll think too much about it. So how do we do an assessment? What kind of tools do we have to assess whether or not somebody's sleeping well and whether or not they're breathing well during their sleep and whatnot? So usually the first step will be an unattended pre-screening assessment. So we send people home with a polysomnogram machine that they can set up on their own, and it's a simple version. So it will have a pulse oximeter, so we can measure oxygenation. It will have a chest belt, so we can see if the chest is moving, if there's actually effort to breathe. It will have some kind of airflow, a nasal cannula. It will have a snore device. It's basically a microphone that allows us to hear whether or not they're snoring or vibration, and an EKG, cardiac rhythm. After that, if we've looked at it and we realize maybe there is something going on, we will have the person come into a sleep center and do an attended polysomnogram. So this will include all of the above, but it will also include an EEG, muscle movement and body position, and we will also possibly institute positive airway pressure like CPAP, BiPAP, and do titrations. So when we look at our man here, this is basically a whole setup for a polysomnogram. At the top, we have the EEG, and I did add a PowerPoint on how to put the EEG electrodes on, so go ahead and review that. You're not going to be responsible for that until you actually do a polysom. It's something you would need to practice with, so if you're going to do any sleep lab rotations, I encourage you to get ahead and practice putting the leads on the head. Okay, so we got the EEG at the top. We will always have a ground electrode, and that is going to be right in the middle of the forehead. We'll have an EOG, so this is an electroocularogram. We're looking at the movement of the eye on the right side, and this will be placed 1 centimeter above the right outer canthus. That's the corner of your eye. And then we'll have one on the left side also, and this will be placed 1 centimeter above the left outer canthus. Then we'll have our pressure, temperature, or flow cannula as a nasal cannula. We'll have a snore, microphone, or vibration recognizer measurer. We will have electrodes on the chin so that we can determine whether or not the person is in REM, recognizing the state of atonia. And the chin EMG is going to be placed midline. One of them will will be midline, 1 centimeter above the inferior edge of the mandible, and 2 centimeters below the inferior edge of the mandible, and 2 centimeters right and left of the midline. So you'll actually have 3 of them there. We'll have EKG, and most of the time it'll just be lead 1, and that's where you can see one on the upper right and upper left chest there. There will be a position sensor that's almost always placed on the chest belt, and that's what you see in purple on the blue chest belt. And then there will be an abdominal belt, a pulse oximeter, and then limb EMGs, right and left. And sometimes we put them on the arms also. And this is when we're looking for leg movement. There are some sleep disorders that are due to periodic leg movements, and we'll talk about that when we start talking about diseases. Okay, so if we've done a polysomnogram and we realize that somebody has sleep disturbances, apneas, etc., then we will definitely talk about lifestyle education and modification. Some people will only need an oral appliance or oral device, and this is something that is fitted to their mouth that will allow their mandible to be pulled forward. Sometimes people have a recessed mandible that causes them to have that obstruction when they go to sleep lying down. If that's not the problem, then and they have apneas, obstructive apnea, CPAP might be an option, but we would test that and titrate that. So in summary, the sleep-wake cycle is a very complicated physiological process that involves the RAS, the SCN, and cholinergic pathways, among others. Sleep disorders can cause chronic disease, and a polysomnography is used to assess sleep disorders. The next few slides represent the modified 1020 system, or EEG, that we use specifically for polysomnography. It doesn't include all of the electrode placements for a full EEG. And I'm not going to read it out loud to you, but please go through it and get an idea of how you might use this on a person. What I would do is get a flexible flexible measuring tape and like a charcoal pencil and do these measurements on somebody else's head. Just follow my step-by-step process, and you'll do great. I will give extra credit for any of you that will do a video showing that you've practiced this 1020 system on another person. So I hope you'll do that. It's kind of fun. You won't be doing that unless you won't be doing this unless you do a polysom rotation, which I hope you do.